Hello, my name is Raymond Alvarez, and I am co-founder of iCore Biologics, as well as an assistant professor in the Division of Infectious Diseases at the Ichan School of Medicine at Mount Sinai. And first, I'd like to thank the organizers for inviting me to speak about some of the things I've been doing over the last several years involving Fc gamma receptor signaling during viral infections. So I think this subject is quite pertinent in the environment that we're currently in. So as you all well know, at the end of last year, there was a SARS CoV-2 pandemic outbreak that currently has infected 24.2 million people worldwide, with 5.8 million people in the US. Currently, the mortality rates are running at 3.5% worldwide and 3.1% in the US. And so the urgency of this pandemic has sent scientists racing to develop vaccines that elicit neutralizing antibody responses. In parallel, other researchers are focusing on developing antibody-based therapeutics that can be passively infused into acutely infected individuals. So I think this has put a spotlight on antibodies, although the emphasis has mostly been placed on the neutralizing capacity of antibodies, whereby the antibody targets the viral antigen that interacts with the host receptor, thereby preventing permissive or infection into permissive target cells. However, the back half of the antibody also has functional properties. So the antibody is a bifunctional molecule whereby the back half, the Fc constant region, mediates non-neutralizing antibody responses. And the way this occurs is that the antibody is able to recognize either viral particles or virus-infected cells. And then the Fc portion, the constant region of the antibody, then interacts with Fc receptors on the surface of innate immune effector cells. This interaction then causes such effector responses as antibody-dependent cellular cytotoxicity, phagocytosis, and cytokine production. So these responses are mediated by a family of Fc receptors, and in particular, the family that recognizes IgG antibodies are Fc gamma receptors. And so what happens is that once an antibody-antigen complex is detected by these Fc receptors, they become cross-linked. And so this family of Fc gamma receptors consists of 6 activating and inhibitory receptors. And because of their different affinities, as well as their ability to both activate and inhibit effector responses, they modulate the immune system in response to pathogens. So there are 2 primary pro-inflammatory Fc gamma receptors, which is Fc gamma 2 and Fc gamma 3, and they mediate a lot of antibody effector functions. And so these effector functions are ADCC, as I mentioned, where an effector population detects an antibody-optimized infected cell and then releases perforin granzyme to kill that cell. Antibody-optimized cells can also be detected by phagocytic cells, which are like macrophages, which end up phagocytosing and killing infected cells. And also, antibody-antigen complexes can be taken up by dendritic cells, the antigen then being processed and presented to the cellular arm of the immune system. So all of this can mediate the expression of cytokines, which further dictates the environment in which an immune response is conducted. And so further regulation of Fc receptor interactions are mediated by the different subclasses of IgG antibodies. And here you can see IgG 1 through 4. And so depending on what subclass is targeting the antigen, you'll get subtle regulation of Fc receptor interactions. Adding to this regulation is the potential glycosylation that occurs in the Fc region of the antibody. And you can see here the variety of glycosylation patterns that can occur, which add to the potential complexity and subtlety of disease regulation. Another variable is cell type. And so you can see here that Fc receptor expression is target cell dependent. So you have quite a variety of different types of effector populations that express different levels of Fc receptors, and their expression can vary depending on tissue localization. So all of these factors contribute to Fc regulation of immune responses. So antibody affinity, subclass, glycosylation, epitope targeting, and avidity, as well as multivalency. So all of these factors contribute to how Fc receptors orchestrate the immune response. So you can see how complex and how subtle the system is in terms of being able to regulate immune responses. So now I'd just like to turn our attention to how Fc receptor signaling has been shown to be involved in viral infections. And so in the context of influenza, we can see here the hemagglutinin envelope protein. It consists of a globular head as well as a stalk region. And researchers have found that anti-hemagglutinin antibodies that are directed against the globular head actually have very efficient neutralization, but they have very poor FcγR receptor activation. This is good for strain-specific responses. So we get seasonal flu, and it responds to that particular strain. However, recent developments have identified antibodies that target the stalk region of hemagglutinin. And what these antibodies do is they also can mediate neutralizing and non-neutralizing responses. However, these non-neutralizing responses can recruit effector cells and mediate more effective immune responses. What's interesting about these anti-stalk antibodies is that they're broadly reactive. So the regions in the stalk are more conserved amongst more strains. So the basis for a universal influenza vaccine is developing around eliciting these anti-stalk broadly reactive antibodies to then generate a universal influenza vaccine. So other evidence of the importance of Fc-gamma receptor signaling in viral infections come from the field of HIV, where the first clinical trial to show protection from the acquisition of HIV also came up with the first immune correlates with protection. And those correlates were not neutralizing antibodies, nor were they cellular responses. What protection did correlate with were high titers of non-neutralizing antibodies, particularly against the V1, V2 region of the HIV envelope, so the crown region of HIV env. And these non-neutralizing antibodies seem to mediate higher ADCC responses against the virus. So further evidence of Fc receptor involvement in the in vivo control of HIV comes from my own work at Mount Sinai, where we examined a cohort of viremic controllers that had viral loads below 1,000 to undetectable levels in comparison to HIV chronically infected patients. And what we observed was that while viremic controllers and chronically infected individuals had similar anti-HIV envelope levels and also similar levels of neutralization activity, what we did find was that they had that HIV controllers had an enhanced capacity to mediate Fc-gamma2 and Fc-gamma3 signaling. So what this is showing is that while the quantitative aspects of antibody responses in both these populations are fairly similar, meaning that the titers are very similar, the qualitative responses seem to be different in that since HIV controllers mediate higher levels of Fc-gamma receptor signaling, this potentially implies that they're mediating, this is contributing to more effective immune responses in vivo. So further evidence of the involvement of Fc receptor involvement in HIV comes from studies examining broadly neutralizing antibodies, where in this case, the broadly neutralizing B12 had its Fc region ablated. And then this antibody was passively infused into macaques and infected with SHIV models of infection. With a wild-type antibody, what you see here is that it maintains both neutralization as well as Fc receptor function, and it pretty much controls virus replication in vivo. However, once you remove the the ability of this antibody to interact with Fc receptors, you then lose this ability to control virus replication in vivo. Another example of this comes from another broadly neutralizing antibody, BNC117, where experiments with this antibody that also knock out Fc receptor function or further optimize it to enhance Fc receptor activity was examined in models, humanized mouse models of HIV infection. And so here we can see that in comparison to wild type, the monoclonal antibodies that maintain neutralizing function but have their Fc interaction ability deleted have higher levels of infection as compared to wild type. And the converse is true for antibodies that have enhanced Fc interaction, where they have a little bit less infection as compared to wild type. This is further corroborated with the viral loads, where the Fc optimized antibody mediates, we see significantly lower levels of viral loads in these animals. So while Fc receptor responses seem to be very important for controlling virus infections in vivo, that's not always the case in every single type of virus infection. In fact, in dengue infection, there are 4 serotypes of dengue infection. And what happens is that you get initial primary infection with one serotype. You then develop an immune response against this serotype. And then the danger then comes in with infection with a secondary serotype of dengue. This reactivates your memory response, but your memory response has been set by your primary infection. So this leads to an inefficient control of the secondary serotype infection. This causes elevations in anti-IgG responses against dengue, which correlates with the severity of disease and dengue hemorrhagic fever. So this caused an initial hypothesis that maybe the antibodies themselves were exacerbating disease. And studies further showed this to be true, where in vitro studies have shown that anti-dengue antibodies mediated enhanced entry and infection of leukocytes, and this is via Fc receptor-mediated uptake. Further, in vivo studies have shown that polyclonal sera from severely infected dengue individuals or monoclonal antibodies mediate pathogenic effects. And further clinical studies have also shown that severe dengue is correlated with enhanced Fcγ3 signaling. In particular, a particular glycosylation state is associated with that. Moreover, enhanced polymorphisms that enhance Fcγ receptor detection of antibodies have further been correlated with disease severity. So we can see how the regulation of Fc receptors may be very important for disease. So we wanted to begin to look at what Fc receptor signaling looked like in SARS CoV-2. And so there have already been immune profiles of mild versus severe patients. And what's been found is that in mild COVID-19, what we see is a initial upregulation of type 1 interferon, which is our body's way of alerting that there is a viral infection occurring. And it activates lots of interferon-stimulated genes, which act to counteract virus replication. So in mild disease, you have this peak of type 1 interferon, which you have some level of replicating virus, which then begins to get controlled. You have lower levels of inflammation, and this coincides with less disease severity. Whereas in severe COVID-19 disease, you have a delay of type 1 interferon responses, where you have earlier phases of uncontrolled virus replication, and 7 to 10 days after the onset of symptoms, you then get escalating inflammation and disease severity. So what these inflammation coincides with is what has been deemed cytokine storms. But really what this is an elevated profile of specific cytokines, mainly TNF, IL-6, and IL-8. These inflammatory cytokines and this increased inflammation and disease severity also coincides with increasing levels of anti-spike IgG. This has led researchers to speculate that potentially ADE is involved in these responses. So other evidence of potential ADE in coronavirus infections comes from SARS-1, where in vitro studies have shown that anti-spike antibodies against SARS-1 mediates higher levels of virus entry into target cells. This is called antibody dependent enhanced infection. Further evidence comes in vivo, where either through passive infusion of anti-spike antibodies or vaccine-induced anti-spike antibodies cause more acute lung injury and cytokine expression in macaque models of infection. The caveat to this, though, is that in monkey models, there is a resolution of infection. And this resolution of infection or the decrease in viral titers coincide with the lung injury phenotype and it resolving as well. A further caveat to the in vitro data here is that while antibodies increase entry into macrophages and monocytes, the population thought to cause the elevated cytokine expression, monocytes and macrophages do not productively become infected with SARS-CoV-1 and SARS-CoV-2. So to begin to kind of clarify whether the involvement of Fc receptor signaling in COVID-19, we gathered a cohort of convalescent individuals. These individuals were antibody positive or antibody negative. There were 28 antibody positive, 20 antibody negative. They were sex and age matched fairly well. Importantly, the time from which there was an onset of symptom in these patients to the time we drew blood was approximately 43 days. That was the median for the population. And this is very important in that 43 days is optimal, since you don't want to get too early of a time point where IgG levels have not peaked yet, and you don't want to get too late of a time point where IgG levels begin to wane. We've also collected blood type data on all of our donors, as well as under any underlying conditions in these individuals. So the first thing that we wanted to do was assess their anti-spike antibody levels, and we used this using a cell-based system whereby we transfected 293Ts with the SARS-CoV-2 spike protein. This then expresses the protein on its surface and allows for detection by donor-derived IgG. This IgG was purified IgG from these convalescent donors. We then come in with an anti-human IgG antibody that has a fluorescence marker on it, and we can then detect the level of antibodies via fluorescence flow cytometry. So we can see here that we have very little background on a negative sample, whereas even with low samples, we get good resolution and detection. This is true also for samples that have intermediate levels of anti-spike binding, as well as high levels of anti-spike binding. So you can see here that the resolution of the assay is very good, where the negative sera is giving us about a 2 log difference between the lowest positive sample that we see here. And we have about a 2 log resolution between the lowest CoV-2 positive sample, antibodies positive sample, and the highest. We can further see a breakdown of this here, where you see the spectrum of anti-spike antibody levels in these convalescent individuals. Importantly, in these 4 individuals with red arrows, they actually tested negative on commercial tests. These tests are normally, these were ELISA-based tests, and they actually said that these individuals were negative. However, we were able to pick them up with our assay, which is evidence that our assay is very sensitive, even detecting low levels. And since we have a 2 log difference between real negative samples and low-level positive samples, we're fairly confident at these results. Also, these individuals did also test PCR positive for SARS-CoV-2. So we also collected symptomology of these infected individuals. And so you can see here the list of symptoms attached to a severity score. We asked each of our donors to self-report the severity of each of these symptoms. What this allowed us to do is come up with a composite total severity score for each individual. While each individual did not have every single one of these symptoms, they had a various array of symptoms, and we could add those scores up to a total composite score. When we did that and compared that to the anti-spike levels, we saw that the cohort pretty much divided into mild versus moderate in scores that were above 45 and below 45. So we then used this to look at anti-spike levels and saw that the anti-spike levels were significantly higher in individuals that had severity scores of above 45. So next, we wanted to assess the level of Fc-gamma receptor signaling in these convalescent individuals. And we again expressed SARS CoV-2 and incubated with donor-derived IgG, except this time we co-cultured these cells with reporter cell lines that expressed Fc receptor cells on their surface. Upon IgG detection, these cells cross-link Fc receptor, causing a signaling to occur and luciferase production. We then can detect luciferase production as a surrogate marker for the level of Fc activation. So in doing this, we see that there is enhanced Fc-gamma 2 and Fc-gamma 3 in convalescent donors with severity scores above 45. Further, when we looked at the level of Fc-gamma signaling in contrast to the level of CoV-2 antibody binding, we found a significant correlation. However, with Fc-gamma 3, this correlation was very striking. It seemed almost like a completely linear correlation where the level of anti-spike antibody very well correlated with the level of Fc-gamma receptor signaling. So what this implies, at least with this small cohort of convalescent individuals, is that Fc-gamma receptor signaling goes up in proportion with the level of anti-spike antibodies. So this was very nice in a set of convalescent individuals. However, we wanted to see how this was reflected in the hospitalized population. And through a collaboration with Saurabh Mahandru of Mount Sinai, we were able to get a handful of hospitalized individuals. We also looked at their level of anti-spike binding. And what you can see here is the level of anti-spike binding in low versus high individuals. And we also see here what it looks like in a severe IgG individual. And you can see how there's a marked elevation in the level of anti-spike antibody binding. When we graph our convalescent cohort in comparison to hospitalized individuals, we see that there's a very significant elevation in anti-spike antibodies in severe and hospitalized individuals. And this is almost a log higher as compared to the mean of our convalescent cohort. When we further divide the level of anti-spike antibody levels in those with severity scores below 45, above 45, and it's compared to the severe and hospitalized individuals, you see this clear stairstep in the level of anti-spike antibody. So we next, having seen this elevation in anti-spike, even beyond our convalescent cohort levels, we wanted to see what the level of Fc-gamma receptor activation was in these patients. And what we observed was with Fc-gamma 2, we did see a trend towards significantly higher Fc-gamma 2, and we did see significantly higher Fc-gamma 3 activation. So it does look as if both anti-spike and Fc-gamma receptor activation correlates with severity of COVID-19. So what we have here is we're left with the question of, are there likely ADE effects in COVID-19? And what we've observed is an increase in the titers of anti-spike antibodies. However, we've seen an increase in Fc-gamma receptor signaling, but it's nowhere near the full difference that we observed in the level of anti-spike antibody binding. So we think that while these in vitro results are very interesting, in vivo results or in vivo studies are warranted, with the important caveat that with HIV and Ebola, there are in vitro correlates that have not panned out in vivo with ADE effects. So in summary, Fc-gamma receptor activation in viral infections, we've seen that efficient Fc-gamma receptor activation correlates with the IgG-mediated control and clearance of infections in vivo. However, with some pathogens such as dengue, enhanced IgG-Fc interactions may actually exacerbate disease. With COVID-19, we've seen that anti-spike antibody levels correlate with disease severity scores in convalescent individuals, and also high levels of Fc-gamma3 and Fc-gamma2 activation correlate with higher anti-spike levels in both convalescent and severe hospitalized individuals. So lastly, I'd just like to take a moment to acknowledge some of the people doing this fantastic work that we've been doing at Ichor Biologics. So the cohort samples were gathered and consented through our company, Ichor Biologics, and this project was spearheaded by our Chief Scientific Officer, Jose Luis Garrido, with help from our great staff of Felipe, Nicole, Sarah, and Sheila. I'd also like to thank our collaborators, Saurabh Mahandru at Mount Sinai for the hospitalized sample, as well as our collaborator, Maria Inés Badia and Mario Calvo, who are currently collecting more samples for us to process. And I'd like to give a special thanks to all of our blood donors. Thank you.